Despite its small size, mosquito is notorious for being the deadliest animal on the planet, responsible for the death of billions. Mosquitoes serve as a vector and host for various diseases, and malaria is one of them. Malaria is one of the most dangerous parasites and possibly the single biggest killer of humans in history. Even though this parasite has been established since hundreds of years ago, there are still various aspects that are not fully understood. To understand a little bit more about this parasite, we made a project titled Identification of Uncharacterized Plasmodium falciparum proteins via in silico analysis. Malaria is one of the most lethal parasitic infections. It is transmitted to humans by the female Anopheles mosquito, which carries the Plasmodium parasites. Anopheles mosquito and the Plasmodium parasites are highly sensitive to any temperature change. Therefore, the constant climate of Indonesia supports the spread of this disease. In addition, Indonesia has relatively high humidity and heavy rainfall seasons, which benefit Anopheles mosquitoes by prolonging their adult longevity. Statistically, Indonesia has over 200,000 malaria cases. Nevertheless, Southeast Asia, which includes Indonesia, is the second most affected region in the world by malaria. There are several types of Plasmodium that infect humans, which are Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, and Plasmodium knowlesi. Plasmodium falciparum is the most severe type of malaria in humans and accounted for half of all malaria infections in 2018. There are three most widely used anti-malarial drugs, which are artemisinin, chloroquine, and piperaquine. However, there are an increasing number of Plasmodium that are resistant toward this current treatment, which complicates the malaria treatment and eradication. Factors that cause persistence of malaria infection is due to mosquito vectors evolving to be insecticide resistant and the lack of effective malaria vaccine, the complexity of life cycle, antigenic variation, and poor understanding of interaction between immune cell and parasite. In this experiment, the method that will be applied relies on several bioinformatic approaches. Bioinformatics is a modern principle that combines computer science, mathematics, and biology for the purpose of solving biological problems. The approaches implemented here are the analysis of genome sequences, protein structure and its function, as well as domain annotation. Genome sequences will aid in the discovery of similar proteins through protein homology, while protein structure can be associated with the protein's function. Domain annotation could also be used to identify protein function from its sequence or structure. Plasmodium has a large number of stage-specific proteins that are involved in motility, maturation, metabolic adjustment, and infectivity. However, there are numerous proteins still uncharacterized until this day. Therefore, identifying the unknown proteins in Plasmodium can be the beginning to solve the complication of this disease by serving as a future therapeutic target. Pandey et al. Studied a conserve with unknown function Plasmodium protein, namely PF3D7 0406000, which is found to be localized in cytoplasm of asexual and sexual stage of Plasmodium parasites. From this example, it can be seen that the uncharacterized Plasmodium proteins have their unique function and benefits that potentially contribute to the parasitic death and elimination in the future. This research aims to identify some uncharacterized Plasmodium falciparum proteins via protein homology to better understand the metabolic activity and discover targets for malaria eradication. The materials needed are 3 uncharacterized Plasmodium falciparum protein sequences and some softwares, namely Fire2, BLAST-P, Interpro, Superfamily Hidden Markov Models, ScanProSite, Ramachandran Plot Server, and Yasara. The first method is to identify the uncharacterized Plasmodium falciparum protein sequences. First, the search term uncharacterized protein Plasmodium falciparum and uncharacterized protein Plasmodium falciparum NCBI were applied to the Microsoft Bing search engine on the 1st of September 2021. Afterward, the uncharacterized protein sequences of Plasmodium falciparum are shown on the resultant page and thus identified. 3 of the 4 uncharacterized Plasmodium falciparum proteins on the sequences from UniProt that are shown on the page are downloaded on PDB format for further analysis. From the protein homology modeling in 3D structure, the FASTA sequences of the 3 P. falciparum uncharacterized proteins were first obtained from UniProt. Each of the FASTA sequences is then imputed into FIRED2 in normal modeling mode. Imputing the email address is highly recommended as the result will be instantly delivered to the email. Imputing the job description is optional, but it can help in identifying the imputed proteins. After 12 to 24 hours, the resultant 3D structure and protein homology model of the uncharacterized proteins are obtained. For validation of the FIRED2 result, uncharacterized protein sequence of P. falciparum and the characterized protein sequence were entered into the BLAST-P to be aligned and validate the similarity. To align 2 sequence, the aligned 2 or more sequence need to be thick. Lastly, click BLAST and wait until the result is presented. To obtain information about the uncharacterized protein domains, Interpro was used by accessing through the website. Firstly, the FASTA format of the 3 uncharacterized protein sequence were inserted into the provided box at the homepage of Interpro. Then, the search button was clicked. After the search was done, the URL was clicked, which redirected into the result page that showed detailed information of the possible family and domains. Superfamily has similar function to Interpro, but it can search for multiple sequences at the same time. It can be accessed through the website. To begin using the website, the FASTA sequence should be inserted into the text box and the search button is clicked. Once the result was shown, all the annotated residue, superfamily, family, and e-values are shown. More detailed information can be found by clicking on the superfamily and family links. Post-translational modification can be done through ScanProSite. Firstly, open ScanProSite website. Then, the FASTA format of the 3 uncharacterized protein sequences are copy and paste into the provided box. Set the setting as default for Submit Protein Sequences column, and for the Select option column, only put text on Exclude Profiles from the scan. For Select Output options and Submit Your Job column, set for Graphical View, and lastly, press on the Start Scan. For protein validation, the 3 uncharacterized protein sequences in PDB format are loaded into the Ramachandran Plot Server with default settings. The residue clustering and protein secondary structures are then analyzed from the Ramachandran plot result. A valid Ramachandran plot should have unallowed residue or outliers of less than 10%, which is shown here in the sentence here that is highlighted in red. For protein visualization and analysis, each of the uncharacterized protein in PDB format is loaded into the downloaded Yasara software. Detailed visualization and analysis of the amino acid residues, protein structure and motifs, and other are then obtained. In conclusion, we expect to identify the structure and function of the 3 Plasmodium falciparum uncharacterized proteins to potentially contribute them as drug targets.